So one of the things that we know about in terms of our study of living organisms is that living organisms, yes, including this little guy right here, this is Foster. He's really not happy right now that I'm holding him. But his fur is made up of a special type of protein, even his skin, and essentially his ability to move his whiskers is made up of predominantly another type of protein that we often look at and call muscle. We put him down for now. We also have things such as homegrown tomatoes, that these are not homegrown because they have a label on them, but these are also a type of organic molecule made up of, shall we say, a bunch of organic molecules put together to form something which we can now identify as a tomato. Even things such as the advertisements that you get in magazines. If I were to put this under a test, such as the use for iodine, I would find that it is made predominantly up of complex carbohydrates. So what we're looking at is that all of these molecules of life is what I'm going to call them, carbohydrates, lipids, proteins, and nucleic acids. They all have an essence of joining together in different ways, depending on the type of bond, and that essentially can form things from cells. So if we have things like we got cells, these cells, remember, were made up of atoms. Cells that work together can ultimately form tissues. Tissues can ultimately form organs. An organ might be something such as a heart. Then ultimately we get an organism, and the organism can be anything from a tree to a fungus to a cockroach to a human being. But it's the arrangement of these types of molecules that will determine the type of organism. And ultimately we're going to see it's literally the nucleic acids, predominantly how the DNA is sequenced, which is going to determine the protein, which will determine the actual organism itself. Carbohydrates are going to be our first line in terms of what we call energy. Every day we need to basically take these carbohydrates and form ATP. ATP, adenosine triphosphate, is the cell's recognizable energy. Same thing as when you go to a different country and you need to convert, let's say, the US dollar into Canadian dollars, or you need to convert the US dollar into euros. All of those are simply because it's that currency of that country. Same thing as ATP, consider that the currency that our cells recognize. And as we saw in the last video, that glucose and fructose, they can combine together by this process of dehydration synthesis to make sucrose. Ultimately, we also know that this can go in reverse, and that if you eat sugar, or sucrose, you will have an enzyme that can break down the glucose and the fructose, and these can consider very similar in terms of isomers of each other. And it's the glucose, this is going to be the one that we're going to convert in terms of cellular respiration, and that will be for a later unit. But recognize that carbohydrates are going to be the first line of our energy, particularly something that we call simple carbohydrates. And we're going to see later on that we're going to use a reagent called Benedict's. And Benedict's is a reagent very similar to how iodine can determine if a starch is complex. Benedict's can determine whether or not a sugar or a carbohydrate is going to be simple. And again, we're going to see that as a color change. I'll give you more on that as the time gets closer. We also have that these carbs here, they're going to have a variety depending on how their structure is. Okay, here we have cellulose, branching. We could also have, that's starch here. Starch, this is going to be cellulose. Ulose. In terms of making up the components of a cell wall of a plant. Starch is different. It's going to be a storage compound commonly found in such as a potato, wheat, rice. But notice the difference is the starch is going to be branched, whereas cellulose are not branched like the starch, and rather they're also informed with another type of chemical called a ligand. Then we got glycogen, which is the storage of sugar in animals, often in liver as well as muscle, but predominantly liver, extra glucose is going to be stored. Now remember, if you go back, glucose is going to be the one type of organic compound that's going to be used as the energy supply, quick energy. So subsequently, the organism, whether it's us, whether it's a grasshopper, whether it's a plant, don't get rid of sugars if there's extra sugars. In other words, we have something called a storage of sugar. So often the storage of sugar can either be in the form of starch, cellulose, or glycogen, depending on the type of organism. The main factor is if we need extra sugar at another time, all of these sugars can ultimately be broken down and back into glucose. Then we have lipids. Lipids are essentially going to be predominantly more structural in terms of their particular function. They could also be used as functional in terms of a backup energy source. For example, brown bears that hibernate, they're going to have something called brown fat. Or we look at migratory birds, they also will have a special type of fat that will allow them to cross those long distance in terms of their migration. Lipids that we can see in terms of our human eye can either be solid, such as a butter. These are considered to be saturated fats. And as we said last time, that means every single carbon is going to be surrounded by hydrogens or other carbons, no double bond. Then we could also see these liquid fats, such as olive oil. The reason is that they're going to have a double bond in between some of the carbons. So lipids can often be insulation factors. They can also be structural factors. They can also be predominantly backup energy. So those are the different ways that lipids are going to be seen in our class. There is one special type of lipid, and that's going to be a phospholipid. Phospholipids are the basis for the cell membrane. They're going to have what is called a polar head, which can be in contact with water, and a nonpolar tail, which is not in contact with water. So we're going to see these things called hydrophobic versus hydrophilic. And the whole way that these are structured is phenomenal in terms of allowing the cell, which is going to be surrounded in water, to still maintain its integrity. We're going to see in a later chapter that these plasma membranes, you may well heard from another class, they are selectively permeable, meaning they can let some things easily into and out of, depending on always the size of a molecule or the charge of a molecule. So that's how they're going to regulate. They cannot regulate something such as a bad toxin, such as carbon monoxide. Carbon monoxide, basically based on its size and charge, can easily enter the cell itself, as can oxygen as well as carbon dioxide. The main purpose for these phospholipids, remember, is the plasma membrane. Another type of fat that you often recognize is that of cholesterol. Cholesterol plays a huge role in terms of a precursor for hormones, particularly testosterone and estrogen. They're very similar in terms of how they are. What you're going to see is this structure right here. This is going to be very similar. This is the predominant basis for what we look at in cholesterol. Cholesterol will also have another role in terms of the plasma membrane and add some fluidity to it so that the plasma membrane can be, shall we say, fluid or semi-squishy, but yet rigid at the same time in order to maintain its integrity and its shape. Proteins are going to be the big ones here. They're going to be muscles, hair, fingernails. They're also going to be enzymes, which you're going to hear a lot about, hemoglobin and antibodies. These are the ways that proteins are going to play a huge role in our bodies. One of the things we're going to look at later on is how proteins are structured from what we call the primary structure, simply the amino acids put together. Secondary structure, amino acids begin to fold in on themselves, whether what we call the alpha helix or the beta sheet. So that's the alpha, that's the beta, and ultimately we're going to get what we call the tertiary structure, and that's how they start to fold in even more on themselves, often with these things called a disulfide bridge. When we get more than one polypeptide joining together, we get what we can consider to be a functional protein such as hemoglobin. We can get protein from legumes, we can get protein from animal fats, we can get protein from egg whites, we can get protein from milk. So there are a variety of sources that we can get protein, and protein can end up being fingernails, hair, antibodies, as we mentioned before. So it's a very diverse type of complex, but ultimately one of the most important ones that we're going to see because, again, that fancy little word that I keep bringing up, proteins also are enzymes. So we can say that all enzymes are proteins, but not all proteins are enzymes. They have a variety of ways that they can have a function in the human organism or a plant organism or a fungus or even a rat. A lab that I want you to take a look at in your book here, it's essentially, if you go back and scroll through page 344, we're going to look at things that can affect how enzymes react. Enzymes are going to be the basis of all chemical reactions, whether it's dehydration, synthesis, or hydrolysis. But we also know that there's a range in which the ultimate or optimum enzyme, in other words, peak activity here of enzyme activity, functions. That could be environmentally determined by the, that's their substrate. We're going to look at how buffers, in other words, how does pH affect the role of enzyme? Some of you might want to figure out, well, how does temperature affect the role of enzyme? So what I want you to do is check out this plan. Not so much here, I'm not even sure why that's even your book right there, except for something about how the enzyme is coded for, but we're going to get to that later on. But we're going to see a couple things. We're going to look at a substrate. And the substrate we're going to look at is going to be peroxide. And the enzyme that we're going to look at is going to be taken from a liver. Yes, liver is another place where this enzyme called catalase resides to break down the peroxides. We're going to see how quickly catalase can break down the peroxides, which, if they're built up in your body in too large a volume, can become toxic. So I want you to read through this, get an idea of what you might want to plan with respect to a lab and how you can determine what type of environment will be beneficial to an enzyme's reaction and what type of environment is not going to be beneficial. And if you're really savvy at here, try to figure out a range that might give you a type of curve that shows you the optimum as well as the low end of the enzyme activities. Nucleic acids, DNA and RNA. We're going to find that DNA communicates to RNA to ultimately do something called transcription, which is essentially write a molecule called mRNA. Some of you might have heard that before. And ultimately this mRNA is going to be translated into the protein, and the protein can have different functions. Both of these are going to be considered nucleic acids because they're going to contain these things called nucleotides. The one difference you're going to notice is the uracil or the thymine from one to the other. The other one is a double helix or two sides. One is only single side. And we're also going to find that there's an evolutionary link between the two here. It is thought that at one point that RNA came first in terms of the evolutionary scheme of life. However, being single-stranded, it really didn't have anything to grab onto. And it also required some of these metals within the clay, how should I say this? Yeah, mud, clay. These metals are going to be the cofactors that held the RNA together. Then somehow, and I don't know how, there's this emergent property that DNA basically ultimately became the genetic code for living organisms, particularly all diploid organisms. Now remember, we still have some viruses, such as retroviruses, the HIV, that use RNA, and those are going to be very sneaky at how they use their RNA to code DNA. But for our main purpose is that DNA and RNA are both nucleic acids. DNA is, shall we say, in command. In terms of a eukaryotic cell, DNA will reside in the nucleus, whereas in a prokaryotic cell, DNA will reside in the nucleoid region. There's another type of nucleic acid, adenosine triphosphate, which I alluded to earlier. It is very similar to the other types of nucleotides, thus we can consider it with respect to that area of a nucleic acid. The difference is that it holds energy. It discretely holds little amounts of energy in what we call these phosphate bonds. Phosphates are going to be negatively charged, so when you try to put two negatives together, you get that very non-stable bond which wants to be broken. Every time it's broken, a little bit of energy is released and a molecule, whether it's a protein within the cell membrane, has been phosphorylated. That now means that energy can be used to manipulate or slightly change the structure of a protein to allow the substrate to fit. Okay. Make sure you read about this in your book. This is a basic overview of it, and we'll talk a little bit more about this in class.